Today we're going to be presenting cell motility and how it affects cancer. We're going to begin by giving background information. My name is Madison Cook.
My name is Tristan France.
I'm Eunice Goodson.
I'm Tamia Davis.
And I'm Tamika Qualls.
Cell motility is the ability for the cell to move spontaneously. A lot of factors play a role in the ability for the cell to move. We have the cytoskeleton, which is composed of microtubules and microfilaments, which is then composed of actin, kinases, and kinesine. Signal pathways help the cell receive information from many receptors to regulate motility. sRNAs cause default in the cell migration and gene expression. Also affects the motility by triggering cancer cells. Cell motility requires protein that makes up cytoskeletons and microtubules, microfilaments, and intermediate filaments. Microtubules are composed of alpha and beta tubulin subunits that polymerize at different rates. Microfilaments are made up of actin, which then twists, forming filaments, which bundles into the cytoskeleton. GNF proteins also form in the actin, known as actin binding proteins. Motor proteins interact between actin and microfilaments and myosin. Kinesine and dynein are motor proteins that control microtubules. Then kinesine moves towards the plus and dynein moves towards the negative. And we have just to describe that. This is basically what I was speaking of earlier. I just was moving faster than PowerPoint. You don't like words anyway. This is the kinesine and the dynein. This is the microtubule, as you can see. The kinesine, it walks along the microtubule with ATP, turning from ADP to the PI movement. And as you can see, this is the dynein moving the opposite direction, which is the negative, and this is the positive. And there you have it.
An important part in cell motility, like she was stating before about how the microfilaments get joined together by the actin coming together. The actin polymerization is a very important key because this is actually the force that actually makes the cell move. This all happens in a kind of complex way, but I'm going to break it down the best that I can. So my understanding is that when F-actin and G-actin filament monomers come together, they form microfilaments. And this happens with the whole ATP hydrolysis, like she was referring to before. And they form like these arrow-like conformations of the G-actin and the F-actin, while when the arrow-like end is the plus end promoting polymerization, and the barb end, which is the negative end, influencing depolymerization. And then, so when ATP hydrolysis comes into action, when the F-actin binds to another F-actin, it cleaves ATP, releasing ADP plus the PI, causing a conformation change in that filament. We'll just say for instance that this side is the minus end, this side is the plus end, since there are actin joining at this side. When the actin binds to the other actin, the F-actin subunits, this causes ATP, like I said before, to do hydrolysis, and it breaks into an ADP plus a PI. And the special thing happens too on this side of the growing microfilament, is that also the PI is released from this side, causing another conformation change that causes the actin not to be able to have actin and actin interaction promoting the polymerization to continue to happen. While at the barb end, it influences depolymerization because it still has the ADP and the PI bound to it. So like I said before, active polymerization is a very key and very important concept in cell motility. But when we come into cell migration, this is what really how the cell moves. It first starts off by extending protrusions. Protrusions are like how the cell moves. Like if I put my arm out here, it's like that's what the cell is doing to make the cell move. And this consists of, the reason why these protrusions happen is because active polymerization is happening, and it influences and it pushes out the actin cytoskeleton, causing a reorganization of that actin. And this is also influenced by a number of signaling molecules that can make this happen. Like she said before about the signaling takes place in how the cell moves. And these can activate different road GTPases and cause active reorganization to happen so the cell can do those protrusions. These protrusions are usually made of filopodia and lamellipodia. And then once that happens, that causes adhesion. Adhesion has to happen after that. It has to connect to the substrate so it can have a tight connection so that it can be able to continue to move. And while this is happening, while this is binding to this part has to be able to retract from that, and then be able to lift up and attach again. And the translocation is like how once it's adhered and once it's binded to the substrate, it can slide across and move. And after that, like I said, the road GTPases come into play, and then they can cause the whole actin reorganization. Because the main thing here of how this all happens is because of actin reorganization, because the actin cytoskeleton of the cell has to be reorganized to be able to make those protrusions or move or make changes. And once the translocation has happened, it's called retraction happens. This is when the adherents here, they pull up from the substrate and allow the cell to move forward. And to tie this all back into cancer, as you know that cancer is a very invasive and very prominent when it's there in the body. Let's try that over again. Cancer is a very invasive type of disease that is brought out by cancer cells getting out of control and migrating different parts of the body. And cell motility is a very important piece in how cancer can become deadly and things like that, because increased motility of these cells obviously can lead to different migrations to other parts of the body, called metastasis. Takes for the activation of the road family GTPases, plays a central role in stimulation, causing the whole protrusions to happen, the whole moving, the migration to happen, like I explained before. And also, microtubules do play a part in that too, just as well as active filaments, because they can actually determine the polarity of where the cell is going to move in that direction.
I'm going to talk about the signaling pathways. It is the RTK ERB2 pathway. Here you have your growth receptor, ERB2. The two of them come together when ligand binds, and it's something called dimerization, but it's really a dimer. So from here you have SRC, and then it's going to bind to SHC. But since ERB2 cannot bind directly to RAS, it's going to recruit SOS, and then it's going to bind to the RAS family protein. All right, which is
Not to cut her off, but to add, except the receptor tyrosine kinase. Once the ligand binds, it had causes the autophosphorylation turning on this pathway.
right. Back to where we were at the bottom, MAP kinase is going to come down here to this, FOSS. And then you have pathways over here. Then this side, you have phospholipase C, beta 1. It's going to come to PIP2, and the PIP2 cleaves DAG and IP3. DAG is the one that remains in the membrane, and then protein kinase is what activates DAG. IP3 is going to trigger the release of calcium, and instead, there's another pathway where calcium binds to comodulin, but instead here, gelsolin is going to come in and bind to the actin protein. Then where PIP2 is over here, you have this type of kinase is going to come in, activate it, and then there's the type of acid that comes in also.
Well, to add to this, to bring it all back, excuse me, into cell motility, gelsolin is, like she said on the slide before, is that actin binding protein that regulates how polymerization can happen. And actually, gelsolin is an ADF, it's an active depolymerization factor, which actually kind of inhibits polymerization from happening. But when that happens, it breaks up like the F-actin units, as you see here, and that initially it increases the way, like I said before, when I was talking about the lamella being able to stretch out of this plasma membrane, extending that. This gives more opportunity for more lamellipodia to exist because there's the actin being broken up. So there's these two pieces can actually bind out of here and do that. So, but this is essential. This pathway is essential because it does do this, increasing motility. Because, like she said before, the ligand binds, growth factor binds. It can activate the the SOS, the RAS family of proteins, and then it can also go down the phospholipase C pathway, and this cleaves PIP2. And to mention, gelsolin is originally attached to PIP2, to the plasma membrane, and the gelsolin gets released from the PIP2 when this pathway is activated. So this whole pathway is really intricate and important to have to allow gelsolin to do its job and allow the F-actin to split up and induce and increase motility by this way. And just to add, DAG is more like a non-polar second messenger, and IP3 is a polar one. Like she said, this one goes usually to the
It usually goes to comodulin, which
yes,
goes
the IP3.
Yes.
IP3 goes to the ER, and that's what triggers the calcium to be released. And the DAG goes into the cell membrane and just does its thing there and activates protein kinase, the protein kinase. So.
I'm going to be talking about siRNAs. I'm going to be talking about siRNAs, and basically siRNAs are small interfering RNAs. They can inhibit migration of cancer cells and the dynamics of gene expression that alter expression of motility-related proteins. This may lead to a dominant negative effect. The ability for siRNAs mediated migration inhibits is assessed by using siRNAs homologous to RAC1, one of the 3 row GTPases that integrates signals with dynamic recognition of the active cytoskeleton.
To point out again that you see that these row GTPases are very critical in actin reorganization. So having to view these siRNAs can really come into great play when there's such thing as cancer getting out of control.
Right. Thank you, Madison. RAC1 siRNA pull, or the SRC, inhibits cause SKOV3 cell migration to be defective by 60 to 70%. siRNAs targeting the MAP4K4 gene on SKLV3 migration are used to show a dominant negative effect. This inhibits range from 55 to 56 relating to control of siRNA transfected cells. The transcription knockdown determined by RT-PCR shows that the range from 64 to 94%. Invasion was inhibited by 76 to 25%, which SI1 and SI2 relative to control transfer cells. Oh, and by the way, this is basically what we wrote in our paper, but we referenced an article that basically shows this in the charts that I'm going to explain next. This test shows that MAP4K knockdown do affect cell invasion. So basically, the results show that tSRNA suppress the expression of intergen subunits of alpha-3 and B1 and MMP9, enhanced it.
So, to help her out with this, these right here are the sRNAs that were added to these enzymes right here in this this research. And right here on the side, relative migration, average viability, you can see that in some places the shading is nearly gone, not even showing gene expression there. So basically showing that
they express more genes when SI1 was added, like they compare from SI1 and SI2. So like when SI1 is added, you show that this amount of gene expression is expressed, or with SI2, this amount of gene is expressed. They're basically comparing from SI1 to SI2 to SI3.
so they're using different types of siRNAs. Yes, their effects on the MAP4K and the rest of what's going to be expressed. So.
And basically, this is basically what we just said in the charts. The results show that the TGF-beta1 enhanced.
But this is also an example.
Right.
This also is an example of how siRNAs are used as well. It's called the tsiRNA. It inhibits the growth factor, the TGF-beta receptor, and T24 bladder cells. This goes back to the RTK receptor system we were referring to before. And basically, an siRNA was used to inhibit basically the ligand from binding to the site to begin the pathway, to begin activating the pathway. And the results they noticed were shown later. And to add to the siRNA, the importance of finding those siRNAs are because maybe they can be a factor that can maybe reduce cancer or subside cancer cells. So that's their importance to cell motility and cancer.
Hello, I will be talking about how gene expression can play a role in cell motility and cancer. To start off, mRNA is a spatial component of gene expression, and non-coding RNA prevents motility by using transcriptional and post-transcriptional to regulate genes factors. I will be talking about the different genes that can affect cell motility in cancer. We have the Acarna gene, we have the autocrine motility factor, and then we have the MLAT1 gene. Okay, so this will be talking about the Acarna gene. The Acarna gene inhibits cancer by altering the actin cytoskeleton. The autocrine motility factor bind to the AMF receptor, so that affects cell motility because it either inhibit or either suppress cancer. And this is MAT1. This is a gene that can enhance motility of cancer by influencing the expression of motility-related genes. Non-coding RNAs play a role in cell motility, and it can also suppress or enhance cancer motility in microRNA's regulatory role is in cleaving RNA and repressing translation that can decrease their motility and migration ability.
To conclude with everything, cell motility is very essential for the movement of cells, for normal cells, but when cancer comes into play, those genes that she was saying, like tumor suppressor genes and oncogenes, they're expressed and they're not being able to cut off, causing cancer to be a very invasive and dangerous disease that proliferates and migrates through the body. And for cells to migrate, they causing stasis to occur. And essentially, the cell has to be able to move to those different parts of the body. And having the actin polymerization happening, and having migration happening, having gene expression happening or not happening, can make this cancer become really dangerous, like I said before. And also the signaling pathways are very important as well, because there are more than just the RTK ones that can actually activate or allow binding proteins to bind to this, chemoattractants, signaling for those cancer cells to go to the tissues. Yeah, those signaling molecules binding them pathways, activating them, releasing active binding proteins that do those things that the normal cells do, but doing it to an extent where it's too much. So I believe that cell motility is essential general for the quality of life of the cell, and it also plays an important part in cancer. Yes.
Thank you.
Thank you and good night.